Hi, my name is Brisa, and today I'll be presenting a biological perspective on the coronavirus, or COVID-19. So what is the coronavirus? Well, it's a group of related viruses that infect both mammals and birds. Now, the news usually uses COVID-19 as a name for the coronavirus. However, it is a respiratory illness that's actually caused by the coronavirus. The disease itself can lead to several respiratory tract infections in the lungs within its human host, and can lead to other mild rhinoviruses such as the common cold. Now, there are 4 known genuses in the coronavirus family, the first 2 being alpha and beta coronavirus. Now, these 2 only infect mammals. However, if we look at gamma coronavirus, this only infects birds, and delta coronavirus can actually infect both birds and mammals. Now, when talking about the coronavirus, it's important to note the cross-species jump. If we look back at the SARS outbreak of 2002 and 2004, the outbreak actually came from the horseshoe bats harboring virus strains that eventually jumped to humans. It's believed that COVID-19 is said to have come about this way, but not fully identified by majority of the people. Now, this is referred to as a novel coronavirus. Novel meaning that it's new and does not resemble anything before known. So it actually doesn't resemble the SARS outbreak virus strain of 2002-2004 because of a different type of spike surface protein that latches onto its host cells. Now, the virus seen today is only one of 7 that have been reported in the past. It can be referred to as aforementioned COVID-19, SARS-CoV-2, and the severe acute respiratory syndrome 2, which is just what SARS represents. Now, if you look here at the bottom, there's a brief timeline, or not necessarily timeline, but the different types of coronaviruses in humans. Moving on to the research done on the coronavirus. Now, UT Austin has actually developed a 3D atomic scale map of the virus molecule, and because of this molecular structure identified, we were able to discover that the coronavirus had a different type of spike protein. Now, if we look at Moderna, which is a biotech company, they released a vaccine for COVID-19, the disease caused by the virus, called mRNA 1273. Now, this is actually one of the first biotech companies that has been able to move into human clinical trials up to date. Now, looking at the Wuhan Institute of Technology, they actually conducted an experiment done on rhesus macaques, or a part of the family, the monkey family, and they actually found them to recover from the SARS-CoV-2 infection. It only gave them a mild illness, and once the researchers re-exposed to them after their initial exposure, the monkeys did not seem to become reinfected. Now, this is essential information when coming about to discovering a new vaccination for the coronavirus, and this is a means for researchers who will be continue to look for evidence to see if humans will react in the same way. Now, the process of viral replication and viral transmission of the coronavirus. So it's important to go over the virus classification. If you pay specific attention to the family portion of the classification, you'll see that the coronavirus is a part of the Coronaviridae family. Now, this particular set of viruses are unique because they have peplomers, or spikes, in the surrounding membrane. Now, if you look at the diagram in the back of the coronavirus, you'll see that surrounding the membrane, it has spike glycoproteins. Now, these are the proteins that will latch onto the host cell and later inject the RNA genome inside of the virus. Now, moving on to the structure of the coronavirus, if you look at the genome, it's actually fairly large, if not one of the largest in the coronavirus family. It's measured from 26 to 32 kilobases. Now, the coronavirus is an enveloped virus, meaning that it does have an additional membrane around the capsid of the virus. Now, the nucleocapsid is helical, meaning it does not have the original icosahedral shape that we see in several other viruses. Rather, it is more like a helix, like a DNA. And if you look at the RNA genome, like aforementioned, the RNA genome is a positive or a single-stranded genome, and of course it is linear. Now, the infection and eclipse period of the coronavirus. Obviously, the coronavirus infects via airborne and human contact methods. However, with the airborne means, we can see that we are not completely certain how long the vapor form of the virus can survive in the air. Now, moving on to the actual infection process of the coronavirus, we see that it uses the spike surface protein mentioned before to bind to a protein cell receptor. Now, this can either be the CEA-CAM or ACE2. And like any virus, there are conformational changes and alterations to the plasma membrane in order to allow the virus to enter the host cell via a receptor-mediated endocytosis. Now, this is basically the cell swallowing the virus. Once the RNA genome is then injected into the virus, it is then encoded by the host cell. Now, the coronavirus utilizes RNA to generate messenger RNA to be transcribed by the host cell. Now, the RNA positive strand or sense strand is then used to make the enzyme RNA polymerase, and the positive RNA strand then becomes replicated to a negative RNA strand. Now, the negative RNA strand is then used by two different methods. The first being either to make subgenomic mRNAs or make more RNA positive strands via DNA replication. Now, going over the first process, using subgenomic RNA, which is a negative strand, is then put through discontinuous transcription to make subgenomic mRNA that's positive, and then once translated, makes different viral proteins. Now, these viral proteins will then be packaged into viral offspring in the virion progeny. Now, the second process uses negative RNA strand or antisense strand to be replicated and then transformed to RNA positive or sense strand that will then be packaged to the viral offspring. Now, both of these components, both the proteins and the strand, are used to actually create the virion progeny. Now, this is a viral offspring that will be released to infect other cells within the host. And it is made via a secretory pathway, meaning that it goes through the roughy arm, then packaged by the Golgi apparatus, and then sent out of the cell via exocytosis. Now, the eclipse period of the coronavirus has not been definite quite yet. However, there has been a study using verocells and the SARS-CoV-2 virus infecting the verocells. What they found was that the verocells had an initial eclipse phase around 24 to 48 hours. Now, this study was conducted by the Institute of Biotechnology in Wuhan and was actually posted to the CDC, the Control Center for Diseases and Prevention of them. And so they also posted this about the incubation period of the COVID-19. They predicted that the incubation period is approximately 5 days. This is because 99% of individuals exuded the symptoms within those 14 days of exposure. However, there were fewer than 2.5% of the infected individuals that showed symptoms only sooner than 2 days after exposure. So we'd see that those 2 days are approximately the eclipse period of the virus. Now, the symptoms and psychological effects of infection and the progression of disease as well. So COVID-19 symptoms include cough, fever, shortness of breath, muscle aches, sore throat, an unexplained loss of taste or smell, diarrhea, headache, and in some very rare cases, COVID-19 can also lead to extremely severe respiratory problems, kidney failure, and even death. 19 is that of lung tissue, specifically two different types of cells. The first being the goblet cell, which produces mucus that allows a wet slimy layer on your respiratory tract on the lining of the alveoli and lung tissue, which is important to keep your lungs moist and allows for a more efficient exchange of oxygen and carbon dioxide in the lungs. Now, the ciliated cells are those that have little hairs on them that wave in an upward direction, and this prevents any nasty or foreign particles that may have gotten into the lungs and can prevent infections from bacteria or viruses. So what the virus does is it kills these cells after infecting them. Now, the dead tissue of these cells then falls into the lungs and can cause not only the symptoms above, but those aligning with pneumonia. Now, funnily enough, the COVID-19 was initially diagnosed as pneumonia rather than the actual virus itself. The immune system can either continue to have those mild symptoms and then get over it, or the immune system can go through something called hyperimmunity. Now, this is when the immune system or immune response over-attacks the foreign bodies and ends up doing more harm in the body by killing healthy tissue under the infected tissue. Now, the virus can also attack kidneys and prevent efficient filtration of blood in the body. Now, about 80% of the population infected with the coronavirus are actually considered mild cases. Now, this is a statistic given to us by Medical News Today, and mild cases are patients who are considered that show no signs of pneumonia whatsoever. So they may have shortness of breath, but not so much so as that's seen with pneumonia. Now, majority of the mortality rate are actually patients with underlying health problems with their immune system, such as cardiovascular diseases, diabetes, chronic respiratory disease, and hypertension. Now, asthma can actually fall under a chronic respiratory disease, and it's actually been seen under patients with asthma that the coronavirus symptoms have actually worsened in them. Now, elderly or people of more age are more prone to immune diseases like diabetes and cardiovascular disease, therefore are more likely to experience those harsher and worsening symptomatic conditions if infected with COVID-19. If we look at a study of the epidemiology in the global age, well, epidemiology study is a science that studies the spread of infections of the coronavirus in this instance, and one of the main goals that epidemiologists have is trying to track patient zero. Now, patient zero can be different in every country. Patient zero could also just mean the first individual to have ever gotten the coronavirus in this instance. Now, the more obvious way of tracking patient zero would be contact tracking, looking at a patient, taking into account their history, the people that they've had contact with, and if any of them exuded those symptoms of the disease being tracked. The next being sequencing technology. Now, sequencing technology is when scientists take a sample of DNA, look at the genome, and see if there are any specific genetic sequence that between patients that are associated with the coronavirus. Now, sequencing technology allows epidemiologists to find similarities in patients with the disease, and it can also determine when they contracted the disease. Now, this is done via viral evolution of the genomes. So, as the virus infects more and more people, there is a possibility of it actually changing as it goes there. So, we can actually determine which patient contracted the virus before other patients using viral evolution. Now, the genome of the coronavirus is 96% identical to that of a bat coronavirus. This suggests that it's possible the bat coronavirus could be the source of the human coronavirus we are seeing today. Now, epidemiologists work together by pairing genomes from different patients with the virus, again using viral evolution to do so. Now, 3 things can come about comparing genomes. A, seeing whether or not it is the same virus. B, how much the virus has evolved within the patient. And C, its origin, both the actual location and the origin, like where it came from. Now, a study conducted in the Gansu province of China, again by the Institution of Biotechnology in Wuhan, was done earlier this year, and the data suggested that most COVID-19 cases that occurred in the Gansu province during the first 6 days were actually imported from Wuhan. Now, the Gansu province is actually pretty expansive, so for a majority of the cases to come from Wuhan suggests that Wuhan, China is the coronavirus epicenter. Now, testing for the coronavirus. There are several methods in order to test an individual for the coronavirus. More so, it's usually clinical and lab work. So lab technicians look for genetic sequences that are specific to the coronavirus and compare those to the human genome sample that they collected. Now, this sample can be collected via a swab test from your nose or throat, a nasal aspirate, which is where they inject a saline solution into your nose, then remove the sample to collect that DNA. And they can also use a tracheal aspirate, which is when a healthcare provider will put a bronchoscope down your mouth and into your lungs, and where a sample of your tissue will be collected. Then there is a sputum test. Now, sputum is a thick mucus that can be coughed up from the lungs. This can also be a symptom that aligns with pneumonia, but can occur with the coronavirus. And then, of course, blood. Now, this has been the main way that people have been tested for the coronavirus. However, several scientists are actually looking towards an antibody test. So labs are trying to develop a sort of exam where they find antibodies created to defend the body from the coronavirus. And so if scientists can isolate these antibodies, they can understand it more and learn to create a vaccine from it. And the interesting part is that the body continues to create these antibodies even after the virus is eliminated via memory and B cells as well. And it can actually track the patient so that the medical professional looking at the sample can see whether there is any reason to believe that there are post-infectionary possibilities within the patient that tested for the coronavirus. Now, testing is only deemed necessary if symptoms are more than mild and exhibit those of pneumonia. The World Health Organization has actually advised those who cannot get tested to stay at home and self-isolate. Obviously, we're all under quarantine, and several federal, state, and local governments have advised citizens not to go to hospitals if symptoms are only mild. Moreover, several institutions have actually gotten pretty creative with how they've tested individuals wanting to test for the coronavirus. One being drive-through testing sites. So the World Health Organization deems it necessary for you to test for the coronavirus if you're having trouble breathing, you have persistent pain or pressure in your chest, you have new confusion or essentially just the inability to move around, and bluish lips or face, or even like bluish, the bluish color in extremities of the body like nose, fingertips, and toes. So treatment and prevention. So something else that could also help treating people with the coronavirus, or just acknowledging or trying to test them to see if they have contracted the coronavirus, is using a COVID checklist posted on the CDC website. They actually connect you with medical experts of the coronavirus to see whether or not you should go and see your doctor. But for those with mild conditions, it's actually recommended that they have self-isolation for at least 14 days. However, if they're still exuding symptoms of the coronavirus for more than 14 days, they recommend staying until those symptoms are gone. Hydration is also essential. Monitoring your symptoms to make sure that they don't get too severe. And if they do get too severe, to call ahead before going to a doctor or any medical center for that matter. And of course, remedies to help with the pain, such as painkillers. Now, for those with severe conditions, isolation wards in hospitals are mainly where they're kept just to prevent contamination to both patients and medical staff working there as well. And they're usually taken on a case-by-case basis. Again, this depends on the different underlying or pre-existing diseases that they may have that have made them more prone to these severe conditions. There is no obsolete or official vaccine that has been created or found or given to the masses quite yet. In fact, the World Health Organization has stated that 70 different types of coronavirus vaccines have been created and are being tested by different organizations around the world. There's already 3 moving on to human trials. This includes the Hong Kong listed CanSino Biologics, the Beijing Institute of Biotechnology, and the Inovio Pharmaceuticals, which is Moderna from Cambridge, Massachusetts. In the new approach of creating the vaccine, pieces of messenger RNA that hold the chemical templates of the spike protein from SARS are injected into human, and then in response to that injection of those molecules, the cells then produce a spike protein that is encoded by mRNA. Now, this triggers the body to that traditional immune response given by traditional vaccines as well. Something else quite talked a lot by the media are the use of antivirals. The 2 popular ones are remdesivir and hydrochloroquine. Now, remdesivir was used originally to treat Ebola in other countries, and hydrochloroquine was originally used to treat rheumatoid arthritis. Now, both inhibit the viral replication by disrupting RNA transcription. As we know, this virus uses an RNA genome, which is why this could prove to be effective. However, there have been several test studies, and it has been proven that it can actually lead to hyperimmunity, again, doing more harm than good. And these antivirals have actually not been approved by the FDA quite yet. So one of the only ways we can truly prevent the spread of the coronavirus is practicing excellent hygiene and social distancing. More mild cases of SARS-CoV-2 have been proven to be the root source and spread of the virus.
My name is Taylor McCormick, and I will be exploring COVID-19 through a chemistry perspective. So currently, the method or procedure being used to test an individual for COVID-19 is a form of a molecular test, and what they do is they swab the back of your nose for at least 10 seconds, and then they send that off to a lab for testing, and this sample goes through what's called a polymerase chain reaction test, and what this does is it detects the genetics of the virus that are present in the swab sample. And in order to have a positive result, the PSR test must detect at least 2 different genes of the virus. If it only has one, then it's inconclusive or negative. And so in the bottom right corner, you can see an example of the polymerase chain reaction test. It's comparing the genetics of the virus with the genetics that are present in the swab. Now, this test is being used in humans, and it's currently pretty limited, but the tests are only being administered to those who are more at risk, such as the elderly, pregnant women, and people with underlying health conditions, and you have to have symptoms of the virus or been in contact with someone who already has COVID-19 in order to be tested, because they're so limited. And the test determines if an individual is affected by analyzing the genes and comparing. Now, there's currently no vaccine or drug to cure or help this virus, but there is some progress that we've seen within scientists and doctors. Now, scientists have begun performing what's called serological tests, and this is a blood test, and it checks your blood for antibodies. This test does take a little bit longer. It takes between 20 and 22 days, and this test is more for people with the disease that have little to no symptoms, and those people with little to no symptoms are more likely to have an antibody to the virus. And there are currently 70 vaccines developing. 3 of those are in a clinical trial stage, which is some pretty good news because we're getting closer to finding something or ruling things out. And even though there's no vaccine, some doctors claim that hydroxychloroquine, it's a drug that's used to treat malaria. They've been administering this to some COVID-19 patients, and they claim that this alleviates the symptoms, and they've seen less severe cases when they administer this drug. Now, there are several chemical interactions that take place when a vaccine is entering our body, and most of these vaccines are full of chemical compounds called adjuvants, and these are chemical compounds that help enhance our body's immune response. So a common one includes aluminum hydroxide, and you'll see the chemical compound of that in the top left corner of the screen. And then some more chemical compounds. These are quite different. They're called stabilizers, and what these do is they make sure that the components of the vaccine, like the antigens and the antibodies that are in the vaccine, stay as effective as possible. And the most common stabilizers are magnesium sulfate and magnesium chloride. Overall, vaccines are filled with antigens, which is a modified version of the virus itself that's used to help your body fight and become immune. But these chemical compounds help really stabilize the vaccine and make sure that your immune system is doing its job. Now, vaccine treatments can be a very long process, and it takes quite a long time to develop a drug and get it approved by the FDA. Now, there's several stages, which include the exploratory stage, which is when you're kind of figuring out what chemical compounds you want to put in this vaccine when you're trying to find an antigen or an antibody. And then after that, you go into a preclinical stage, and this is when you're really developing the vaccine and getting it stabilized. And then if you get your vaccine stabilized, it'll go into a clinical development stage. And this is the stage that 3 vaccines are in right now. It's a clinical trial stage. They're trying it on some patients to see the reactions that it have, if it's effective, the side effects, all of that. And then you go into your regulatory review and approval. And this is the part where the FDA has to approve your drug, has to see the side effects, weigh out the pros and cons, and then if it's approved, it'll go into manufacturing and quality control. Now, normally vaccines can take up to 15 years to be perfected and approved, but in a pandemic, the process is usually spread up. So currently, it has taken us around 3 months to develop some possible vaccines, and normally it'll take 20 months to be in the developmental stage. And it is possible that this outbreak will end before a drug is developed. The virus doesn't strive very well in heat, so it's possible that during the summer we could see this virus just disappear due to natural effects, and we will still be developing the drug, but it won't be necessary at that time. Now, how do medicines and drugs attack a virus, and what components of the virus are necessary to find a vaccine? So what medicines and drugs do to attack a virus is very differently from normal antibiotics. So they don't kill the virus directly. What they do is they provoke your body's immune system to react. And the drugs and vaccines usually introduce the body to a modified version of the virus. That way, if you're infected, your body will be able to recognize and react. Now, in order to find a vaccine, it's necessary to study the genetic compound of a virus and create a similar version that can be used as or that contains an antibody. And it's also necessary to have the chemical compounds to aid this process within your own body, those chemical compounds being the adjuvants and the stabilizers.
Hello, my name is Nireyda Rodriguez, and I will be presenting the physics perspective on this COVID-19 pandemic PowerPoint presentation. To begin, we have manufacturing and delivery of medical equipment. So the medical devices used to diagnose or treat COVID-19 include diagnostics tests, sterilizers, ventilators, respirators, and personal protective equipment, including things such as masks, gowns, gloves, protective clothing, face shields, goggles, and face masks. Next, we have the medical device packaging. So the manufacturer must be able to properly seal a package that is to remain sterile and maintain the aesthetics of the product. The design of the package should be sufficiently robust in order to withstand all types of climates and any physical hazards that the product itself may experience. Lastly, the package must maintain its integrity over time. Next, we have transport. So transportation is a key part of the Defense Department's contributions to the whole of government's efforts in response to the COVID-19 pandemic. Next, we have storage. So distributors must ensure that storage and transport comply with the conditions set by the manufacturer. Temperature always needs to be monitored to make sure that the appropriate conditions are maintained. And lastly, we have delivery. So a medical device travels distances, whether they be large or small, until reaching a storage unit and provider. So through delivery, a product is kept under strong surveillance, making sure appropriate conditions are being maintained, such as the temperature. And it is also seen that there's an increase in the use of air delivery to provide immediate response for relief efforts. Next, we have the materials needed for manufacture. So these materials include metals. So the usage of metal material that resists absorbing or binding with proteins, viruses, and other biological substances that can inhibit its function. Next, we have ceramics. So ceramics play a large role in manufacturing medical devices for being good insulators and being able to be molded to small sizes. Next, we have polymers. Polymers are lightweight, can have excellent flexibility, and are generally inexpensive. Next, we have composites, and this is one of the newest materials being utilized for medical devices. Lastly, we have biomaterials. So biomaterials is a type of material that interacts with the biological systems within the body. So how they are manufactured, medical device makers strive to be efficient and produce quickly, especially because of the growth in the pandemic itself. And there is also a large use of assembly lines, which will be further analyzed later. So where they are manufactured, most medical supplies still come from overseas, while close to home efforts are smaller scale and just getting underway. For example, the Abbott Laboratories. The Abbott plant is in Scarborough, Maine. Moreover, 63 manufacturers, which represent 72 facilities, are located in China, and companies all over America are reaching out to help produce medical devices and provide materials. For example, the Ford company has been producing full face shields for medical responders. And lastly, the assembly. So assembly is a large portion in manufacturing medical devices. Each workstation has a specific set of instructions with color codes, pictures, and descriptions as to what to do and how to proceed in order to achieve an efficient, reliable, and sterile product that is ready to be used by health professionals, scientists, researchers, etc. Following this, we have the description of the physics involved with ventilators. So to begin, ventilators are machines that help patients take breaths if they are unable to do so on their own. So due to respiratory infection, a person affected experiences difficulty breathing and therefore is put to use a ventilator while in the hospital while having COVID-19. It is a bedside machine that connects to a person's airway through tubes, again, helping the patient breathe if they are unable to do so on their own. So how they work. A ventilator mechanically helps pump oxygen into the patient's body. The air flows through a tube that goes down a patient's mouth and down their windpipe. The ventilator also may breathe out for you if the patient decides that that is what they want. The ventilator can be set to take a certain number of breaths for the patient per minute. And on the right side, we have a picture. So first, pressure at point B is equivalent to the alveolar pressure and is determined by the volume inflating the alveoli divided by the compliance of the alveoli plus the baseline pressure, also known as PEEP. And the pressure at point A, which is equivalent to airway pressure measured by the ventilator, is the sum of the product of flow and resistance due to the tube and the pressure at point B. Next, flow, volume, and pressure are the variables, while resistance and compliance are constants. And lastly, flow equals volume divided by time. So here are some recommended improvements for the future of ventilators. So to begin with, the first ventilator was invented in 1928 by Forrest Morden Bird and was also known as the iron lung. The first uses of ventilators were between the 1930s and 1950s to help patients whose breathing muscles had been paralyzed by polio. Since then, there have been many changes due to advances in technology. In the future, it is expected to improve ventilators. To do so, it is recommended to improve on the operator interference to better monitor status of the patient and ventilator, It can also be recommended to better the patient circuit because of a piece of plastic tubing connected to the patient's that is subject to filling with condensate. Lastly, increasing the intelligence of the ventilator to better define, measure, and interpret feedback signals. Next, what technology is being utilized to research the COVID-19 virus? Here are some of the technology that is being utilized, such as applications that collect data to track the spread of the virus. Technology is largely being used to measure the progression of the disease and ensures healthcare centers have access to the supplies they urgently need. Next, we see the use of 3D printers. So 3D printer ventilators for hospitals that have become essential for providing treatment to patients. Next, we see NextStrain. This is an open-sourced project that provides data, sequencing, and visualizations. It is able to show the evolution of pathogens like coronavirus, information that helps epidemiologists understand how it evolves in different countries, along with any possible mutations that can change its nature. And lastly, we see the Abcelera. And the Abcelera is a biotechnology company that is using a machine learning model to develop therapies based on the antibodies from patients who have recovered from COVID-19. Their use of artificial intelligence technology allows for the analysis of more than 5 million immune cells as the search for those that are able to produce antibodies that help patients recover. Next, how does this technology work and how does it assist scientists and doctors? Well, the first piece of technology we will see is the 3D printers, and 3D printers are being used to produce ventilators. So the manufacturer will use polymers in order to continue with a 3D printer that will result in various products, including ventilators. The next product we see are ventilators themselves. So they mechanically help pump oxygen into a patient's body. The air flows through a tube that goes into the mouth of the patient and down their windpipe. Next, we see NextStrain providing data, sequencing, and visualizations to create data on the evolution of pathogens that will in turn display possible mutations. And lastly, we see Abcelera, which is using antibodies from patients who have recovered from COVID-19 to analyze immune cells. So why is it beneficial to scientists and doctors? Well, overall, the goal of a doctor is to save a life. Many lives. A ventilator is able to aid the doctor by providing the patient with a piece of technology that will help them advance in the fight to live. Scientists and researchers are able to use these forms of technology to gather new information, analyze information provided to them, and get closer to a relief and a vaccination. Next, we see a virtual model done by scientists and engineers to mimic this pandemic. So at the University of Texas at Austin, we see this type of research being conducted. So researchers from the University of Texas at Austin remain on campus working on the research on the COVID-19 pandemic. They created a 3D image of the coronavirus, leading them to a potential vaccine that is being tested in Seattle. So the picture on the left is a 3D atomic scale map or molecular structure of the 2019 nCoV spike protein. The protein takes on two different shapes called conformations, one before it infects a host cell and another during infection. The structure represents the protein before it's infected a cell called the perfusion conformation. And next, we see that the molecule the team at the University of Texas at Austin produced and obtained a structure for that represents the extracellular portion of the spike protein. This is enough to elicit an immune response in people and thus serves as a vaccine that is being tested in Seattle. And these are our sources. Thank you so much for listening to our presentation and have a lovely day.